I just wanted to welcome everyone again to our second Units of Life seminar series. As I mentioned in our first time together, we launched this seminar series out of a recognition that in our broad community, we wanted a forum in which we could focus more on sort of deeper mechanistic studies in biology in a sort of hypothesis-driven manner, and to bring speakers from both within as well as outside our community to help inform some of our perspectives in mechanistic cell biology. And of course, Units of Life, cells being the units of life, we thought was a appropriate name for this new seminar series. And as with all experiments, Jillian Shaw, who's been a remarkable designer of these series behind the scenes, and I would love to hear your feedback as we go along and hopefully improve the series to fit better with what our community would like to see or needs to hear. So I just wanted to also bring your attention to the fact that we have a great lineup for the spring series. And we are actually currently planning to move to in-person meetings in the spring, starting on January 5th with Helen Hobbs, who's visiting us from UT Southwestern. Helen is, of course, extremely well known, and it will be wonderful to be in person and be able to welcome Helen and hear from her and her remarkable work. So I hope that we will be able to reconvene in person at that time. I've heard from many that this will be a welcome change to what we have all gotten almost too used to, these Zoom meetings. And then we have a terrific lineup for the rest of the spring, and it's both investigators from our community here as well as people from outside our community. I think it will be a great treat, and I hope to see you all there. So without further ado, I actually wanted to introduce our speaker for today. It's a very special pleasure and honor to have Dr. Tobias Walther here today. Toby is a professor of molecular metabolism at the Harvard School of Public Health. He's also a professor of cell biology at Harvard Medical School. We are lucky to call him an associate member here at the Broad, and he's importantly an investigator of the Howard Hughes Medical Institute as well. I understand that Toby received his PhD at the EMBL in Germany, and then completed postdoctoral studies with Peter Walther at UCSF. He then returned to Germany at the Max Planck, and then went to Yale as a faculty member until in 2014, I think it's fair to say we were lucky to have recruited him to Harvard, where he started a joint laboratory with his longtime collaborator, Robert Farisi. And so the Farisi-Walther lab basically studies membrane lipids, lipid metabolism, and cell biological mechanisms of homeostasis. And I think we're in for a treat today, learning more about their recent work in this area. So again, I'm very grateful, Toby, for agreeing to come speak with us today, and very much looking forward to your talk. I'd like to remind everyone to please use the Q&A at the bottom to ask your questions, and I will do my very best to moderate. Thank you all. Thanks, Toby. Over to you.
Thank you so much, Anna, for a great introduction. It's a pleasure to be here, of course. I wish it was in person, and I hope we can do that again soon. And before I get started, as Anna mentioned, I want to highlight that all the work that I'm going to present today is the result of a scientific partnership between myself and Bob Faris, and we've been co-running a lab now for the last, well, I don't know, 8 years and counting, and before that we're close collaborators. And here's a little bit of a reference to some of the things we've learned. But of course, we don't do things ourselves that often, or at least not alone. And I want to particularly highlight today work from a couple of terrific laboratory members, particularly Shuiwu Sui, Henning Arlt, Jaeyoon Chung, and Nicholas Mayard. And they, of course, built on discoveries that were made by other people in the lab and elsewhere. And we have great collaborators throughout the community. And today I'm going to particularly highlight work that was done together with Mafu Liao at the medical school, Jung-Kyu Park at Wayne State, Joel Goodman at UT Southwestern. And then we have great collaborators for more theoretical work that I'll mention briefly, and particularly Si-Young Kim and Greg Vogt's group at the University of Chicago, and Jessica Swanson, who was there for a while, but now has her own group at the University of Utah. Now, what I'm going to talk to you today about is a fundamental principle of life, and it has to do with the fact that biological systems really exist because of energy flow through those systems, and there's constant energy intake and a need to expand energy. But for many systems, the available energy, as well as the need to expand this energy, is not always constant. And as a consequence, biological systems generally have evolved mechanisms to at least temporarily store metabolic energy. And by and large, they do so in the form of lipids. So this cartoon here is drawn roughly to scale for a healthy human to show how much we store metabolic energy in the form of lipids and to a lesser extent, carbohydrates. Now, here's an example where this is particularly uncoupled temporarily. So this is a sockeye salmon in British Columbia. And what has been done in the 50s is basically measure the body composition of these animals as they migrate from the ocean here up to their spawning grounds. And you can see that the protein stays the same, but in this case, the storage is really temporarily uncoupled from, the intake is uncoupled from the need to expand energy. So as these salmons migrate up the river, they lose a lot of that fat mass, that lipid. Now, the major form in which bodies store the lipids is called triacylglycerol. And it's triacyl because it has 3 acyl chains that are esterified to a glycerol backbone. And because this molecule is a lipid without a head group, really, it is a neutral lipid. And you all know, of course, what happens when you take such a neutral lipid, an oil, and mix it with water. The 2 phases don't intermix with each other. And instead, the neutral lipid forms oil droplets in water. This is probably one of the reasons, actually, why this molecule is so widely used as a storage molecule, because excluding water allows it to extremely densely pack metabolic energy in those carbohydrates. Now, you can recapitulate this, of course, in vitro. And here's a simple experiment on my backyard porch where I mixed some oil with water in a cocktail glass. And what you, of course, see is that the 2 don't mix and that there's an interface. And this generates a fundamental problem for cells because pretty much all the biochemistry and the life happens in the aqueous phase. And the energy storage is now decoupled in the oil phase. And so cells have evolved mechanisms to allow to deal with that oil phase, to deposit energy into that phase and withdraw it. And they do that all on an interface that can be very problematic. Some of you may remember old ways to extract DNA, where we used phase separation to extract DNA, and then all the proteins would denature at this interface between the oil and the water. So there are really special mechanisms required to deal with this. Thus, the presence of lipid droplets converts cells to emulsions. In these emulsions, the cytosol now forms a continuous phase, and the lipid droplets form a dispersed phase. And for cells to be able to deal with that oil phase, emulsifiers are needed that keep the droplets separated and accessible and prevent proteins from just crashing out of their surface. Now, this has important physical consequences. One of them is that the emulsion diffracts light. And as a consequence, lipid droplets were among the earliest discovered organelles. This is shown here by a German pathologist, Richard Altmann, who drew this beautiful picture of a frog liver cell in which you can see all these lipid droplets accumulating. This, incidentally, is the same person who coined the term nucleic acid and is also credited with the discovery of mitochondria. Now, we now know that lipid droplets are highly unusual organelles. You can see on the schematic here that they are composed of a core of an oil phase, mostly triacylglycerols, but also other neutral lipids, such as sterol esters, retinol esters, waxes, depending on cell type, that is separated from the rest of the cytosol by a monolayer of phospholipids. This is very different from all the other organelles that always have a bilayer membrane around them. And into this are specific proteins embedded. And it was a key discovery by Dean Landas and others that there are really specific proteins that are only found on lipid droplets and that really define this organelle. Lipid droplets within cells also interact with many other cellular structures. For instance, you can see on this micrograph on the left here in a hepatocyte or hepatoma cell that they appear to form contacts with mitochondria, and they're often found in close association with the endoplasmic reticulum. I mentioned to you that the proteins are really important, and work from many laboratories, including ours, has identified sort of the proteome of lipid droplets that includes several hundred specific proteins, again, depending a bit on the cell type that's being analyzed. These proteins include enzymes, for instance, as you might expect, for depositing and withdrawing fat from the lipid droplet, but they also have there's a whole slew of proteins for which we either don't know what their function is at all, or for which we do not know why they are on lipid droplets. And there's just a couple highlighted here. And to make that information more accessible, we've actually worked with our colleagues at the Broad and particularly developed a knowledge portal that allows the integration and access of data from proteomic, gene perturbation, and other screens to get integrated. And you're happy to check this out here and see whether some of your favorite proteins might be there. Now, in multicellular organisms, the storage of lipids is often uncoupled in specific tissues known as adipose tissue from the rest of the body. And you can see here that in adipose tissue, the lipid droplet is a unilocular, one giant droplet that fills out essentially the whole cell, with the rest of the cytoplasm squeezed here to the site in these sort of darker regions. But importantly, pretty much every other cell type in the human body appears to have the potential to form lipid droplets. For instance, after a meal, the intestine temporarily forms lipid droplets when fatty acids come in, and before they're being able to be shipped out. The liver is very important for energy homeostasis and can temporarily store fatty acids, for instance, derived from adipose tissue during fasting. And lipid droplets can also be found in other places where we know a little bit less of what they're doing, for instance, in the brain. And lipid droplets are part of really fascinating physiology that we have very little understanding of. For instance, milk, of course, contains a lot of fat, and lipid droplets are the components of that milk fat, but we have really no clear understanding of how the shedding process that forms the milk actually occurs. Now, during these processes, what happens is that the fatty acids are essentially catabolized to neutral lipids and packaged into this more inert form of a lipid droplet organelle. And this removes these excess fatty acids from the cell where they could be bioactive. Now, I mentioned this equilibrium, but of course, in modern society, very often the intake and the output of energy from human body is not exactly in balance. And as a consequence, often we now observe that there is obesity, which is the massive overaccumulation of fat and adipose tissue. And what is thought to happen under these conditions is that the adipose tissue and other cells start to store fatty acids and make large numbers of lipid droplets, but eventually the capacity to do so in cells is overwhelmed, which then leads to the accumulation of lipid metabolites, which can trigger stress in the cell, for instance, in the ER membrane, and cells then either adapt or eventually die, leading to pathology. Depending on where this happens, different pathologies are associated with obesity, most notably, for instance, fatty liver disease, where fat accumulates in the liver, often linked to obesity, which is coupled to something known as the metabolic syndrome, characterized by diabetes, insulin resistance, etc. Lipid droplets also store other lipids, such as sterol esters, and in that context, they're particularly linked to cardiovascular disease. In addition, we now know that there is many genetic defects that interfere with the body's ability to store lipids. And this has very similar consequences because now lipid droplets cannot form effectively. And as a consequence, similarly, lipid metabolites accumulate and become toxic. We now know that there is, particularly, for instance, in adipose tissue, there are mutations that compromise the ability to store lipids, and these manifest as so-called lipodystrophies. And although that person on the right here might look relatively normal to you, he is severely metabolically sick and has an inability to store fat and adipose tissue, and as a consequence, there's severe metabolic disease and insulin resistance, diabetes, etc. Depending on where proteins that might be affected here are expressed normally, lipid droplet proteins or genes linked to lipid droplet biology show up in many congenital diseases. And here's just a couple of examples here that have been characterized by many laboratories. Now, today, I'm going to talk about three topics. And I'm first going to speak about our current understanding how lipid droplets actually are formed. What is their biogenesis? I will then mention to you what our current understanding is of how how proteins actually target specifically the surface of lipid droplets. And in the final part of this, I'm going to share with you a sort of a news story where we investigate, of course, how the lipids are mobilized from lipid droplet, which is important if you want to use the energy stored in them and the fatty acids for energy and membrane generation. Now, for the first part, how do lipid droplets form? Now, the textbook understanding from just a few years ago is roughly this. Lipid droplets magically appear somewhere in the ER membrane and form randomly a lens somewhere. That lens then magically buds towards the cytoplasm, and somewhere in this process, specific proteins are acquired. And of course, this is not really satisfactory, and we really want to understand how this process works. And so first and foremost, of course, in order for this to happen, cells need to have the capacity to synthesize triacylglycerols or triglycerides. This occurs particularly through two distinct enzymes, and they're named after the activity that they're catalyzed. These activities were discovered by Eugene Kennedy and colleagues in the 1960s and are named after the reactions that they catalyze: diacylglycerol acetyltransferase 1 and diacylglycerol acetyltransferase 2. And when Bob discovered the actual genes encoding these enzymes in the late 90s, it quickly became clear that those enzymes really only share the name, and DGAT1 and 2 are very, very different. DGAT1 is a polytopic membrane protein of the ER membrane, and DGAT2 has this apparent anchor with a hairpin, but they really share no sequence homology to each other. They also have different cell biology. DGAT1 is always in the ER, and you can see here on the left in a Drosophila cell in red that the protein stains this reticular pattern throughout the cell, whereas in a Drosophila cell, DGAT2 also localizes predominantly in what appear to be specific lipid droplets, and you can see sort of a subset here stained in red. Now, how do these enzymes actually work? We recently had a bit of a breakthrough here by solving the cryo-EM structure of DGAT1. And when Shui Wu did this, he found this remarkable butterfly-shaped dimer that is localized in the endoplasmic reticulum membrane. And when we analyzed particularly one of the two subunits of this dimer, we found that the catalytic center for this reaction is deeply buried within the plane of the membrane and is accessible from two sides: from the cytoplasmic side here through a tunnel, and then from the membrane side through a lateral gate. And so what we think, based on a series of structures as well as biochemical experiments, how this reaction occurs is that fatty acyl-CoAs are inserted from the cytosol or the cytoplasmic leaflet of the ER into the active site. Then a catalytic histidine catalyzes the esterification to a diacylglycerol that enters from the side tunnel from the plane of the membrane, in this reaction excluding water and therefore allowing efficient esterification. And when esterification occurs, the triacylglycerol is then directly released into the plane of the membrane. We know much less about how DGAT2 works. We do not know where its substrates come from, where the triglyceride goes, and how this localization around lipid droplets occurs. You can see here that the active site, based on AlphaFold 2 prediction, this was one of the proteins that the AlphaFold people highlighted in their original publication, appears to be on the cytoplasmic side, and we on others often find the protein associated with lipid droplets, but how that occurs and how it works is not clear yet. Now, I mentioned to you that triglyceride storage is most often found as a hallmark, or accumulation is most often a hallmark of obesity. And one of the most critical unmet medical needs in this area is fatty liver disease, which now affects 1 in 4 people worldwide. This is not only true in the Western world, but really is a global problem. Now, because this disease is characterized by an accumulation of triacylglycerol, pretty much every major pharma company at some point developed inhibitors for DGAT1. Here mentioned a couple of them, with the idea to block the triglyceride accumulation and thereby prevent disease. But when we found humans that have mutations, loss of function mutations in the DGAT1 protein, in collaboration with Harland Winter and Mark Daly at MGH, these people had severe congenital diarrheal syndromes with a protein-losing enteropathy and a fat-dependent diarrhea. And it turns out that when humans lack DGAT1, particularly after a meal in the gut, fatty acids come in, but DGAT1 is the only DGAT expressed in the human gut. And as a consequence, enterocytes cannot handle the fatty acid influx. And instead, our current model is that toxic lipids accumulate that then lead to cell death, and that leads to then eventually the degeneration of a lot of those enterocytes. And based on this knowledge, Nina Glachowski in the lab has been able to develop a treatment strategy for those patients. Consistent with this, all these drug candidates here have failed, mostly due to severe GI side effects, which is of course genetic, consistent with the human genetic. So then, of course, this brings up the question, so is all hope lost here for treating fatty liver disease with therapy focused on DGATs? We don't think so, because when we generated animals that lack DGAT2 specifically in the liver, this protected the liver from the accumulation of fat without any harmful consequences to the animal. And consistent with this, Pfizer now has completed successful phase 1 trials with markedly reduced liver fat for DGAT2 inhibitors, and they've moved these compounds forward also in combination with other inhibitors. And so how does this work? Is there no accumulation of toxic lipids? Well, it turns out that for DGAT2 inhibition, there is a feedback system that specifically downregulates a transcription factor known as SRABP. And this transcription factor is key for lipid synthesis in the liver. And so what happens effectively if you inhibit DGAT2 is you not only block the synthesis of triglyceride, but you also feed back onto the synthesis of more fatty acids, and thus downregulate the whole pathway. And as a consequence, this presumably prevents the accumulation of toxic liver. And in accordance with this, Pfizer is now also trying to use these inhibitors in combination with inhibitors of fatty acid synthesis, or early upstream steps thereof. And we're now, of course, very keen on understanding really what is the mechanism of this error here that's highlighted here. But now back to lipid droplet formation. So once the triglycerides are synthesized, how do they actually get packaged into a lipid droplet? Key to this process appears to be a protein known as Sapin. This was first identified as mutated in patients with the severe congenital lipodystrophy I've shown here before, which is associated with mental retardation. And Sapin deficiency perturbs lipid droplets in many systems, and this has been analyzed in a large number of different laboratories. In our lab, this had shown up in a large systematic screen in human macrophage cells where BSCL2 encoding Sapin was one of the strongest hits. And you can see on the cellular level, if we take fibroblasts from patients with a Sapin deficiency, lipid droplets do not form normally, but instead of this sort of normal array, there is now a couple of large lipid droplets, and then there's sort of all this sort of schmutz here in the background of the cell. And so how does this phenotype arise? Well, it turns out that this has to do with the sort of the formation of lipid droplets. Here, there is a Drosophila model system, and you can see that in a wild-type cell, the cell has a few pre-existing lipid droplets, and then a few lipid droplets are formed in what appears to be an organized process. But when cells do not have Sapin, there's a few of these large droplets, but now lipid droplet formation does not occur normally, but instead you get all this sort of snowstorm of this little grainy lipid signal that appears to not able to form a functional lipid droplet in the ER. And we now know from work of many laboratories that this leads to sort of a whole dysfunctional droplet that acquires sort of the wrong set of proteins and also is not functioning in lipolysis. Now, how does this protein Sapin actually work? We were quite intrigued to learn that when we tag the protein at its endogenous locus with a fluorophore, that it forms these foci shown here in green that localize to the endoplasmic reticulum where lipid droplets form. And you can see that there's a zip around here. And this is true in evolutionarily conserved fashion. It's shown here on the left for Drosophila cells and on the right for human cells. Now, what are these foci? How does this protein actually form these foci? So Sapin itself is a small protein. It's roughly 50 kilodalton, has two transmembrane domains, a luminal domain, and short variable extensions in the cytoplasm at the N and C terminus. But when we started to analyze the biochemical properties using cryo-electron microscopy, we found that the protein assembles into a large complex, consistent with biochemical data, for instance, from Joel Goodman's lab. And you can see here that the whole protein makes a large complex that sits in the endoplasmic reticulum membrane, and where there is windows in the plane of the membrane to the interior of this whole cage-like structure in the ER. And when we tilt this now and you look at it from the cytosolic side, you see that the complex appears to be closed from the cytoplasm. And when we start taking away the transmembrane domains and focus on the luminal domains, you can see there's sort of this ring assembly of the whole complex in the ER membrane. Again, this assembly of Sapin is evolutionarily conserved. We now have structures from our lab and Ying Yan's lab for the yeast, fly, and human complex. And you see that there's a very similar arrangement, although somewhat peculiarly, the number of subunits in the different species that form the most stable complex are variable. Within those luminal domains, there's two features that stand out. A beta sandwich fold domain, shown here on the side, that is reminiscent of lipid binding proteins, and the hydrophobic helix that each subunit projects to the center of the complex. And if we turn this on the side, you can see that this hydrophobic helix is also projected upwards towards the plane of the membrane. And we quickly hypothesized that these hydrophobic helices help organize the triglyceride and help the phase separation to form a lipid droplet. But there were sort of two things that really bugged us about this. And the first thing is we could never really isolate triglyceride with the complex when we purified it. And you can make up excuses for that, such as, there's detergents around and what have you. The second thing that bothered us a little bit is that particularly when Jae-Yun Jung in the lab analyzed the sequence of these hydrophobic helices, she noticed that they're not only conserved evolutionarily in their hydrophobic character, but also in their specific sequence. So Jae-Yun hypothesized that maybe they bind another protein, and she designed an experiment, and here I just show you one of many, where she generated a Sapin that doesn't have these hydrophobic helices, and compared in a proteomic pull-down experiment what the full-length Sapin might interact with, which is missing when you don't have this the hydrophobic helices. And she's done many control experiments that don't have time to show you, but just show you here in this discovery experiment, there was really one protein and one protein only that interacted only with the Sapin that had the hydrophobic helix. And it was a poorly characterized protein called TMEM159 that we've since renamed lipid droplet assembly protein. And when we tagged this protein at its endogenous locus in cells that also express Sapin, tagged at its endogenous locus, and we engineered in a third fluorophore to be able to follow lipid droplet formation, this is a lipid droplet surface protein, perilipin, we found that lipid droplets really only form at foci in the ER that have both Sapin and LDAF. This is quantitated here, where we follow where lipid droplets form, and we found really that lipid droplets only form on sites in the ER where both proteins are present. We can, sorry, there's something wrong with the slide, but we can generate an experiment to ask, okay, does this really determine where lipid droplets form? And what Jae-Yun did here is an elegant way where she used a chemical dimerizer to bring LDAF and with its Sapin to the plasma membrane, right under the plasma membrane, and anchor the ER onto those sites. And you can see first here, so this works when you add a chemical dimerizer, everything goes into a contact site between the plasma membrane and the ER. And as you might expect from the fact that the two proteins interact, that brings with it Sapin. And really then, only then can we observe that lipid droplets form right under in this contact site of the ER and the plasma membrane right there. Again, consistent with the idea that this complex really catalyzes where lipid droplets form. Now, during the formation, if we wait longer, something fascinating happens that we don't fully understand yet. Sapin and LDAF start out together in a complex, and you can see this here with overlapping colors in the endogenous proteins in green and magenta. And then as lipid droplets form, the complex sort of buds out, and the LDAF decorates the surface of lipid droplets, whereas Sapin stays back at the neck of the ER and lipid droplets. And this has since also been corroborated by, for instance, Elena Ikon and other labs. If we do not have LDAF, as you might expect, lipid droplet formation is compromised and is less efficient. In particular, the cells are able to make less lipid droplets for the same amount of TG, consistent with the idea that really this complex helps to package the triglyceride into a lipid droplet. And so biochemically, I told you already that if you just purify sapin to apparent homogeneity from cells, you do not find any triglyceride associated with it. But if you start purifying both proteins, sapins and LDAF, we now see some triglyceride associated with this complex. And that is greatly boosted if we induce lipid droplet formation by the addition of exogenous fatty acids. And we know this is triglyceride because where it runs, but also because it is sensitive to inclusion of inhibitors of the DGATs. And structurally, as these lipid droplets form in the complex, by cryo-EM, we can observe that a flat micelle, seen here on the left now, where there's the luminal domain in blue and the micelle shown as a flat micelle, starts to bulge out from the ER membrane and forms a sort of a curved micelle, which we believe is sort of the earliest intermediate of lipid droplet formation that we can observe. So if you put all of this together, our current model for the initial droplet formation is that at the endoplasmic reticulum, DGAT enzymes synthesize triacylglycerol from diacylglycerol and fatty acids and release it into the ER membrane. The complex of Sapin and LDAF1 then provides a space where those triglyceride molecules can interact with each other rather than with the phospholipid side chains, effectively catalyzing the phase transition and allowing the lipid droplet to form a lens within this complex. We suspect, based on the cryo-EM data that we have, that that complex can open, because we see two different conformations, one of them more open, and that allows the lipid droplet to egress from the ER at those complex sites. And in the process, sort of the lid is blown off and the LDAF starts to decorate the newly forming lipid droplet. Now, in this process, at some points, the proteins target lipid droplets. And this is particularly important because these proteins are really the way for the cell to deal with the phase of oil. Generally, we now believe that there's two pathways how you can access the surface of lipid droplets. And one pathway, proteins are translated in the cytoplasm and then bind directly to the surface of lipid droplets through generally sequences that have the propensity to form amphipathic helices, in which there is an alpha helix in which one side is hydrophobic and one side is hydrophilic. The second pathway starts with the translation of proteins and insertion into endoplasmic reticulum, and then somehow the transport from the ER to the surface of lipid droplets. And we call these pathways operationally cytold and ertold for cytoplasm to lipid droplet and ER to lipid droplet targeting. Now, for the cytold pathway, the mystery really is, like, how do these proteins specifically bind the surface of lipid droplets rather than the massively more abundant endoplasmic reticulum? And mind you, for other organelles, there are specific landmarks such as phosphoinositides on the plasma membrane or the endosome, but those are not present on the lipid droplet. There's also no specific insertion machinery known, such as the translocon on mitochondria or the ER. So how does this actually happen? From a combination of biophysical experiments and simulations, we now believe that the lipid droplet surface is quite special. The presence of triacylglycerol under the surface pushes apart the phospholipid chains to some degree and allows for some of the triglycerides to access and come out to the surface and generate a sort of a rougher surface. This rougher surface is characterized by these little packing defects where water can access directly to the hydrophobic phase. And we've shown in simulations and biochemical or biophysical experiment that these defects are larger, more persistent, and more frequent than you would observe on other membranes. We can recapitulate this binding in vitro using giant unilamellar vesicles. So these are giant bilayer membranes into which we incorporate triglyceride, generating a system in which a monolayer surface is contiguous with a bilayer surface. And then we can add cargoes such as the cytold proteins and ask where do they bind. And you can see here in this experiment that cytold cargo proteins, such as a peptide derived from the CCT metabolic enzyme, accumulates on the monolayer here, but not on, to a much lesser degree, on the bilayer membrane. We know we can manipulate the system and manipulate particularly the surface tension by providing either pressure or suction onto it and imaging what happens to protein binding. And just as an example, you can see here when we increase the number of packing defects by increasing the surface tension, more protein binds to the surface. On the side of the protein, it strikes us that the hydrophobic face of these proteins that bind lipid droplets have many large hydrophobic residues, such as tryptophans, for instance. And we think that these are particularly important because in this in vitro system, we can take such a helix that doesn't have large hydrophobics and it doesn't bind. We engineer in a number of these large hydrophobics and now it binds specifically to the monolayer. We can take a natural version of such a peptide that does have a larger hydrophobics and mutate them to small hydrophobics and we lose binding. And this is also true in cells. And so what we now think is that what occurs here is that the large hydrophobic residues insert to the packing defects and adsorb to the surface that way. That this initial adsorption then drives the folding of the amphipatic helix into a alpha helix to satisfy the backbone needs for hydrogen bonding. And then this essentially makes this process irreversible because now the free energy to unfold this protein would be very, very high. Now we're still learning how this process participates in physiology, but I want to tell you sort of one sideline story very briefly of a surprise here, how Cyto is used in an unexpected regulatory paradigm. I mentioned to you that we, and in this case particularly Niklas Meyer, did a lot of proteomics on lipid droplets and cataloged which are the proteins that are there. And when we did this, we find often, so the plin proteins are already mentioned briefly and many other well-known proteins. But when Niklas did this, he also found this protein here, MLX. And so this turns out is a transcription factor. And it was also a major hit in the screen I had shown you before. Of course, when I first saw this and Bob first saw this, at least I was very dismissive. I thought like, it's a transcription factor. It's not a lipid droplet protein. There's no way. There's got to be a contaminant. But Niklas, to his credit, prevailed in really characterizing this. And he was particularly motivated because this transcription factor is part of a family that includes MLX, MLX-IP, also known as MONDOs, and particularly CHRBP. And these are major regulators of metabolism, and particularly the response of cells to glucose and converting glucose to lipids. And here's just an example that showed us that this protein indeed really does bind to lipid droplets in pretty much every system we've looked at. And consistent with this was also found in other people's proteomic experiments using APEX and other technologies, for instance, such as done by James Oldsman at UC Berkeley. We mapped that the protein, this transcription factor, actually contains one of these amphipatic helices used by Cyto and binds lipid droplets that way. And you can see here that a sequence with such an amphipatic helix is both necessary and sufficient to bind lipid droplets. And when we mutate individual residues in that sequence, in this amphipatic helix sequence, we lose binding. Now, one of the major targets in many cells of this transcription factor is a protein called TXNP, which is an alpha-arrestin characterized by Lou Cantley's laboratory that is very important for the response of cells to glucose. And so we used this as a reporter and first found that the expression of TXNP is a faithful indicator of MLX activity. If we knock out MLX, here shown on the right, pretty much expression of this TXNP protein is gone. And you can also see when we add glucose to the system, in wild type cells, there is about a threefold induction. But when cells do not have lipid droplets, this is vastly amplified. So now there appears to be more of the transcription factor available to do this regulation because there are no droplets in these cells that are treated with DGAT inhibitors. Conversely, if we drive lipid droplet formation, we see that actually more of the transcription factor is bound to the surface of lipid droplets and the response to glucose is blunted. So this leads us to a model in which Cyto here is used to sequester transcription factor on lipid droplets. In cells that do not have lipid droplets, this transcription factor locates to the nucleus and responds to glucose. And it actually switches on genes that are required for the conversion of glucose to fatty acids. But when cells already have a lot of fatty acids in the bank in the form of lipid droplets, the transcription factor is sequestered by erythritol on cytol on the surface of lipid droplets, preventing its localization to the nucleus, and as a consequence, preventing its activation of gene transcription. So we think this is an elegant way how the lipid storage response operates and coordinates the physiological response according to what already is in the lipid droplet bank in terms of fatty acids. Now, how about that second pathway I mentioned, erythritol? In this pathway, proteins are initially inserted into the ER, and then they reach the lipid droplet. But even we know that proteins don't just hop in and out of membranes and then swim through the cytoplasm to their new destination. So how does this actually work? Here you can see the erythritol pathway in action. This is a metabolic enzyme known as GPAD4, catalyzing the first step in triglyceride synthesis or glycerolipid synthesis. And you can see that in cells that do not have lipid droplets, this protein here tacked with a red fluorophore localizes to the ER membrane, tacked in green. But after lipid droplets have formed, the two signals segregate, and now the enzyme is only found in lipid droplets, really, and leaving back the ER. So how does this actually occur? Well, you might think, well, lipid droplets form in the ER, the protein's in the ER, of course, the protein just goes on when lipid droplets form. Well, it turns out that Jion Sung in the lab found that this is not the case because the Sapin and LDAF complex really prevents most proteins from accessing the lipid droplet surface during their formation. And, if you just think about it, this complex has roughly 66 transmembrane domains right at the neck where the lipid droplet comes out of the ER. It's not hard to imagine that most proteins can't go through that. Instead, something really magic happens, and very much later during formation, not during initial formation, a specific subset of lipid droplets appears to acquire the possibility to gain these proteins from the ER and then do so very, very rapidly. And we think that this occurs through the establishment of a second independent bridge between the ER and the lipid droplet. And we call this the Lateair pathway, which we think is used by the majority of proteins that come from the ER to the lipid droplet surface. And we think that this might include very important proteins for metabolic disease, such as HSD17B13, that some of you may have heard of. On a structural level, we can observe these bridges with fluorescent microscopy, and also here with electron tomograms, where you see a ribosome-studded ER in physical continuity with the lipid droplet surface. And this is here just a false colored image of the same. We, of course, are very interested in how these are actually established. And in data that I don't have time to show you today, Jion recently completed a genome-wide screen identifying the factors that are required for this targeting and found that there is specific members of the ER exit sites, as well as tethers and fusion machinery that are required to form the bridge between the ER and the lipid droplet surface, and subsequently targeting of GPAD4. This also allowed us to identify many of the proteins that use this pathway, but we still need to really work out of how this reaction occurs. Now, of course, just because there's such a connection formed doesn't solve the whole problem. Just because there is a bridge doesn't necessarily mean everybody has to run across it. How is it that proteins accumulate on the surface of lipid droplets rather than equilibrating between the ER and lipid droplets? And again, the ER is vastly more abundant, which would mean most of the protein would stay back, and you wouldn't get that image that I've showed you before where all the proteins are on the surface of lipid droplets. Now, this seems to be a sequence feature of these cargoes. Many of them contain this long stretches of hydrophobic helices or of hydrophobic sequence, which have uncharacteristic placement of some positively charged residues in them. And Maria Jesus Elarte in the lab found that when you scramble this sequence, the protein is still inserted into the ER, but now this sort of minimal fragment that we can recapitulate erythritol is not capable anymore to accumulate on the surface of lipid droplets. And she found that particularly these positively charged residues, as well as a couple of large hydrophobic residues in the center of this, are needed to allow for the accumulation of these erythritol cargoes on the surface of lipid droplets. We don't really understand how these features work together, but based on molecular dynamic simulations, our current model posits that positively charged residues anchor the hairpin sequence to both sides of the bilayer. Now, this forces in turn large hydrophobics into the center of the bilayer, which is energetically unfavorable because now they disturb the order of the bilayer instead of being at the interface. When the proteins localize to the lipid droplets, we think that they all swim up and the hydropositives and the large hydrophobics now are on the surface, which we know generates a conformation that's energetically favorable. And because of this energy difference, we think that the proteins eventually end up migrating to the surface and accumulating on lipid droplets. Now, in the last few minutes, I want to switch gears a little bit and tell you about, like, once you've made a lipid droplet, the whole thing is only really good if you can also withdraw things from them. All the good things in the account really only matter if there's a way to get things out of the ATM here. And generally, to allow for getting fatty acids out of a lipid droplet for the generation of metabolic energy or to synthesize membrane, there are two pathways. One is known as lipolysis, in which surface lipases on the lipid droplet directly cleave the triglycerides and free fatty acids. Beautiful work, for instance, from the laboratory of Rudi Zechner has characterized how this process operates, primarily in adipocytes. It's definitely also there in many other cell types, but there it is less well understood. In addition, in a second pathway, a lipid droplet or a part of a lipid droplet can be engulfed by an autophagosome in autophagy, and then the whole thing gets degraded in the lysosome, freeing fatty acids from there. Now, this lipophagy or lipid autophagy was postulated and characterized more than a decade ago when Ana Maria Cuervo's lab made mutations or depleted core autophagy machinery proteins and found that as a consequence, lipids accumulate. But of course, there's many ways this could happen, and all of them might be direct. And a key question for the field now has become is, like, how does this actually operate mechanistically? And what is the specific machinery that targets the lipid droplet? And Jeon Chung in the lab really had a recent breakthrough in this, I believe, when she started to characterize a protein known as Spartan, tagging it here with a fluorophore at its endogenous locus. She found that this protein, Spartan, localizes to a subset of lipid droplets, stained here with BODIPI, that is distinct from those that acquire the perilipin 3. And again, all of this is with endogenous tagging. And of course, this was really puzzling to us. And Jeon quickly wondered, okay, what is special about these lipid droplets? And she analyzed sort of their relationship with other cellular organelles and quickly found that the lipid droplets that have Spartan also co-localize with autophagy machinery components such as LC3A and lysosomal machinery as LAMP1. You can see this here in this merge. Molecularly, this protein Spartan consists of a number of domains, particularly a somewhat historically poorly named ubiquitin binding region or UBR domain and a senescence domain based on a plant screen. And in data that I don't really have time to show you today, what we found is that the senescence domain actually contains an array of sequences that can form amphipathic helices and that are sufficient and required for the protein to bind to lipid droplets. We've also found that the UBR domain specifically interacts with members of the autophagy machinery and the LC3 proteins, thus positioning Spartaning ideally to become an adapter between the lipid droplet and the autophagic machinery. To test whether this protein is really important for the autophagic degradation of lipid droplets, we needed an assay. And Jeon adapted a system that's been used by others, and in our neck of the woods, particularly Wade Harper, where she generated a live drop. This is an embedded lipid droplet protein fused to a chemofluorophore. This fluorophore has some peculiar properties, and it changes its excitation spectra depending on the pH. So that in the cytoplasm, it is mostly green fluorescence or excited by green, whereas when it is in a lysosome, where the pH is much lower, it shifts to a red excitation maximum. And you can see here that when you have sort of wild type cells and withdraw fatty acids and thus prime the cells to metabolize their lipid droplets, many of these lipid droplets end up in the more acidic environment of the lysosome, and the color shifts. But when you do not have Spartan, this doesn't really happen anymore, really indicating that the protein might function in lipid droplet autophagy. Now, consistent with this, if you do not have Spartan in cells, the cells have more triglyceride. And they don't have more triglyceride because they make more, because the synthesis rate is the same, but because they hydrolyze it less. And to the same extent, roughly, they are compromised in the mobilization of triglyceride that what you observe when you mess with the autophagic machinery. And I'm only showing you a subset of these experiments here. Now, I haven't told you yet what Spartan is and how it's got its funky name. So Spartan is also known as SPG20. So, and this was discovered as mutated in a spastic paraplegia, which is an autosomal recessive disease characterized by progressive muscle weakness, a developmental delay, and also small stature. And consistent with this human pathology, Spartan is highly expressed in the brain, both in neurons and glial cells. So of course, this prompted us to ask whether or not Spartan and with that lipid droplet autophagy, lipophagy might be particularly important in neurons. And so Jaehyun set up a system in which we can generate motor neurons from wild type or Spartan deficient iPS cells. And what she found is that really when Spartan is gone, lipid droplets start to appear. You can see here in this magnification that normal neurons, these eye neurons in vitro, have very little lipid droplets, but when you do not have Spartan, they start to accumulate triglyceride. To test this in the context of physiology, Jaehyun collaborated with Jaehyun Park and Wayne State, who generated a model in which we injected either controls or a dominant negative Spartan that Jaehyun had generated into the two hemispheres of a mouse brain to serve as control and treatment groups. And what we found is that only when we interfere with Spartan function, neurons start to accumulate lipid droplets. And we can see that the lipid droplets are decorated with this sort of dominant negative Spartan. And consistent with this, when we isolated these brain regions and analyzed them by mass spectrometry, mass spectrometry-driven lipidomics, we find that triglycerides massively accumulate in the neurons that are where we interfere with Spartan function, and really only there. Whereas all the other lipids that we detect essentially stay the same. So this leads us to this working model where Spartan functions in lipid autophagy, and particularly in neurons, and serves as an adapter for the degradation of triglycerides. Now, it is not lost on us that normal neurons don't have a lot of triglyceride, and that this is really weird. This is not the first organ I've had thought about in triglyceride metabolism, but this is really where the biology leads us now. And there's a couple of really important other facts I didn't mention yet. So Sapin, that I mentioned to you before, is also known as SPG17, and different mutations in this protein also lead to a spastic paraplegia. I told you about Spartan as SPG20 in lipophagy. And what I hadn't told you yet is that there's also a lipase, DDHD2, initially discovered by Ben Cravat, and which we then characterized with him, which is also known as SPG54, which is also linked to, in this case, the degradation of lipid droplets through lipolysis. So together, this really leads us to ask, okay, what is the role of triglyceride flux in neurons? And I think this is something that Jaehyun will hopefully work out. Now, in summary, I've told you today how DGAT synthesizes or releases triglyceride directly into the ER membrane, how we currently think the Sapin and the LDAC, the lipid droplet assembly complex, work together to mediate the initial lipid droplet formation, and how this restricts proteins from lipid droplets. That there are two distinct pathways onto lipid droplets, at least, and that we've discovered a specific adapter for lipid droplet autophagy. I've also tried throughout the talk to highlight how the central features of cell biology, as we unravel them, are really important to understand metabolic disease, and that understanding these fundamental processes of cell biology can relatively quickly lead to therapeutic approaches for these diseases. Now, I'm both happy and embarrassed about the progress we made. I'm happy because I think we make some progress, but I'm embarrassed because there's so many open questions. And I want to leave you with just that we really don't understand most things rather than we do. We don't understand DGAT2. We don't really understand how lipid droplets form for other lipids. We don't really know exactly how the LDAC does its magic for phase transition, how are these bridges actually formed that I mentioned, how they're regulated, can proteins go both ways, what happens when droplets shrink, when they're digested, how do you get to some lipid droplets? I showed you one example where this happens. And then, also for lipolysis and lipophagy, I think the relationship between the two is not very well understood. So I think there's lots and lots of things to do. With that, I want to quickly acknowledge some of the other people who've contributed. I already mentioned the people in our lab that have been working on this most recently. And I've also acknowledged most of the collaboration partners that we've had. And of course, this built on a number of people who've now left the lab and who've contributed to the story. First and foremost, I want to thank my scientific partner, Bob Faris, who's just been a fantastic partner in crime and who would have loved to be here, but had a family thing that he really needed to do today. And I also want to thank our funding sources, particularly the National Institutes of Health, Pfizer, and the Howard Hughes Medical Institute. And I'll thank you for all your attention, and thank you very much.
Toby, thank you. That was a tour de force on lipid droplet biology. That was spectacular. There are some questions coming in. One of the people in the audience wants to know how stochastic versus regulated is the heterogeneity in LDs intracellularly?
That's a good question. I think I don't think we know that completely. There are a number of, there's many factors, right? Lipid droplets are easy to see, right? And that's but there are many, many, many factors that impact them. The key variables are how, the speed of their formation, the different phospholipids available for the surface of lipid droplets, the remodeling of those phospholipids, some of the proteins that coat this emulsion. And so, in many, many cells, I think a number of these parameters are very tightly regulated. We know that. And some of them are probably stochastic. And then I don't know what the balance is and how much you get from one versus the other. And I hope that answers the questions to some degree. In addition, there are some cell types where, for instance, I showed the adipocyte. There's a machinery that was characterized particularly by Peng Li, where really lipid droplets are coalescing and form this unilocular droplet. That's done with a specific machinery known as the FSP27 proteins and SIDEC proteins.
And can I just follow up on that question? What about, and I noticed in a lot of your experiments, you're basically using oleic acid, if I understood correctly, as sort of a stand-in for the supply of fatty acid. What about varying the type of fatty acid that would be available? Do you know of any heterogeneity that comes from that? Or?
Yeah, I mean, there is, there is a whole very large body of classic biochemistry that has studied particularly the substrate preferences of the enzymes of the Kennedy pathway culminating in the DGATs. DGATs are particularly good in using unsaturated fatty acids, and they do like a diacylglycerol that has one saturated, one unsaturated fatty acids. They can take lots of other things. And you appear to be making a lot of lipid droplets. But, for instance, palmitate is quite a poor substrate for many enzymes in the pathway, and as a consequence is one of the more lipotoxic species in vitro. And you can model that. But we see sort of the same requirements once the triglyceride is formed for the machinery there. I hope that answers it a little bit.
Yeah, fantastic. There's several more questions. Is the role of Spartan in LD autophagy brain specific, or can these findings be translated to other tissues?
Yeah, that's a great question. I don't know the answer.
Mhm.
I also, I really don't know the answer. The protein is expressed in other places, and one would expect if it's there that it might do the same thing, but we don't know, and we've only tested it so far in the context of neurons. I also, just to say that, because that often comes up as well, is like, what's specific about motor neurons? Why would
Right,
motor neurons
right.
be particularly susceptible? I think this is a question that's true for many of these susceptibility loci. You know, we have a different project working on frontotemporal dementia, where a very specific set of neurons is first affected. And similar to the Spartan case, we really don't know why particular subsets of neurons are more susceptible, and whether other neurons are, and then whether other cells are. My suspicion is it might play a role also in other tissues, but you know, that hasn't been analyzed. As far as the patients are concerned, this is a spastic paraplegias come in a pure and a complex form. This is a complex form, so they have many other phenotypes, so to speak. They have many other pathologies, and this would argue that this process plays other roles. But I should also say that Spartan has also been shown, or has been claimed to play a role in all kinds of other processes. We have been having a hard time finding evidence for some of the other things with our work, but it's also possible that in addition it does something else. So I don't know.
Makes sense. Another question, are the same dynamics taking place for lipid droplets that surround and contact mitochondria? Is there something unique about these specific lipid droplets?
We have not worked on this. There's a paper from Orai Shirihai at UCLA that has separated lipid droplet populations based on whether or not they're in close proximity or associated with lipid droplets, and actually has, in that paper, it is claimed that the mitochondria are different depending on whether they are sort of associated with lipid droplets or not. I think similar to the lipophagy. So that's interesting. I don't really know much besides what's published, and we haven't done any experiment here. I think similar to this lipid droplet autophagy field, in the absence of mechanism and machinery that specifically links the droplet to mitochondria, all these questions are super hard to ask, right? Because just you can come up with different reasons why you might purify things differently. And I think what's really missing here, and is a great opportunity for someone to do something, is like the specific machinery that links mitochondria to lipid droplets. I think that's a big outstanding question. There is a perilipin that somehow seems to be important for this, but it's not sufficient to do this. And it's not mechanistically clear. It's called perilipin 5. But so we don't know as a field.
Very interesting. I think we touched a little on this, and you mentioned it in your unanswered questions, but someone in the audience wants to know if you have any insight on how cholesterols are deposited in lipid droplets.
Yeah, again, not so much from our own work. The first step in this reaction is ACAT, and actually Bob had worked on this enzyme, ACAT 1 and 2, and they form the cholesterol ester analogously to a DGAT reaction, and there's a structure also, and so this works very similarly. We have no reason to believe that the formation would be different, but it's not fully tested. There is something interesting. So if you just take cholesterol, and particularly cholesterol ester at room temperature, of course, it's not liquid. If it's uniform, at least it would be a solid. Consistent with that, initially Gunther Daum and then more recently Yulia Mohammed have observed that cholesterol ester-rich droplets can undergo a phase transition on their surface, where the cholesterol esters form a liquid crystalline phase right under the monolayer, and it is now quite obvious that that these then could attract different proteins, and we have published some of those differences, and more recently also Mike Henney at UT Southwestern has found that this is differences. But I think that's fascinating. We don't really know. The screen we did, the genome-wide screen we did that I just showed sort of glancing by, used human macrophages or human THP1-like macrophage-like cells, and we used an LDL, a modified LDL that would both store cholesterol ester and triglycerides And we have a bunch of hits there that are maybe more geared towards the cholesterol ester storage, but haven't done much with them yet.
Yep, more work to do. A very interesting question that I haven't thought about. Is there a relationship between the lipid droplet size and toxicity that is observed?
Yeah, I'm sure there is a relationship. I often feel that, you know, the lipid droplet size is like, it integrates so many different things that I'm not 100% sure how predictive it is, because you can get to a larger lipid droplet in probably 200 different ways. And some of them might be really coupled very tightly to toxicity in a causal way, and the other ones are not. One thing, you know, I think for most part, people now think that the accumulation of triglycerides per se is not toxic, but that it is a hallmark of when there's a lot of flux through the pathway and that there's lipotoxic intermediates. One thing that gave me pause in this is when a former graduate student that then joined Matthias Mann's lab at the Max Planck Institute did a study in high-fat diet animals. So her name is Natalie Kramer. And she found that in high-fat diet animals, when there's massive steatosis, the lipid droplets sort of start to accumulate proteins of the secretory pathway of the Golgi right on the surface of lipid droplets and sequester them. And this is done in an unbiased way in these animals. And that as a consequence, secretion from the hepatocyte was lowered. And that would potentially give you a futile cycle where there's more triglyceride that leads to a downregulation of secretion. And of course, the main thing the liver secretes is triglyceride. So then you get more steatosis, and that would be a pathological effect. And in that case, a larger number and a bigger surface of this would be very important. And I encourage you to look at Natalie's paper if you're interested in that. So it's interesting study.
Very cool. I just want to acknowledge that we have gone over time, but there's one or two more questions. So I'm just happy if you're happy to go ahead with a couple more questions. And of course, understanding that some in our audience may not be able to stay, but there is another question which ties into something I wanted to ask. Is there an insight from lipid droplet physiology into what components of diet, saturated fats, fructose, ethanol, fiber, cause the epidemic of obesity and fatty liver disease? I guess going, you know, at the level of the whole organism, what can we glean?
Yeah, I think it's also an interesting question. And I hope you keep that question in your mind when you talk to Helen Hobbs in just a few weeks, because she's done some very, some really impressive work on the genetics, but it's also, cross-talking to diet. And I wish I could just bump this question to some of my colleagues in the School of Public Health, such as Frank Hu or and Walter Willett, who've spent their whole careers on this question. I think it is somewhat clear that saturated fatty acids are more toxic than non-saturated fatty acids. High levels of cholesterol intake are really bad, and this is for different reasons. This has to do with cardiovascular risk and the accumulation of foam cells. So this is a tough one to answer, because at some level, there's an incredible amount of knowledge in this, in literally 60 years of nutritional epidemiology that I'm not qualified to answer. And at the same time, I don't think we know really causally very well for how these things are linked. But certainly alcohol leads to alcoholic steatosis, and that has some of the same and some different consequences. And saturated fatty acids particularly appear to be toxic in the diet. And things like the Mediterranean diet are attempts to combat that through nutrition. But have a look at Frank Hu and Walter Willett's work. I mean, that's quite amazing, actually, what they've been able to do.
That's fantastic. And just the last thing that I wanted to ask, going into the mechanism, I was very interested in this beautiful sort of toggle for the MLX transcription factor between being sequestered in the lipid droplet versus not. But then it got me thinking, does that mean that there's a kind of ceiling to how much triglyceride you can sort of store in your lipid droplets? Because presumably, let's say if you were to feed more glucose, for example, but now you're unable to convert that to fat, does that mean, what happens to that, right? Like, you know what I'm saying? That there should be there's no MLX to deal with the sugar. So what happens?
Yeah, that, and this is another source of embarrassment. I remember vividly that when Bob and I first started work together, and I was a postdoc and he was on sabbatical, we had coffee together many times, and we're talking about these things. And one of the first questions that we struck on is like, okay, you can take 20 different mammalian cells and feed them with fatty acids, and they all make a different amount of triglyceride, and then they stop. And they don't stop because you could change the media and things like that. It's not like they run out of something. They just stop. There's something happening. And then if you force it with other fatty acids, then they die, right? But it's not the same amount of triglyceride. Every cell makes different. And obviously an adipocyte can make tremendous amounts of triglyceride and be very well functioning. And at some point, even that cell might be overwhelmed. So what determines the capacity of a cell to store triglyceride, and what are sort of the ceiling for that, and what are regulatory mechanisms, to my knowledge, is not well understood at all, and is probably a super important question. Also, Nicholas has started his own lab and is, I think, very interested in that, and he's super good. So look out for what he's going to find. He's one of the smarter people I know. So I think that's a super fascinating question. And again, I wish I had something really definitive to say.
Yeah. Well, you've already been incredibly illuminating. This has been a tremendous conversation, and evident in all the questions and the engagement that we had from the audience. So I'm very grateful, Toby, for a great talk and for spending all this time with us today.
Thank you,
thanks
Anna.
Again.